Good afternoon. Welcome, everyone. Thank you for joining us for the broadcast of the Monarch Butterfly Conservation Series. Today's topic is Monarch Research and Advanced Topics. My name is Rhonda Miller, and I work for the U.S. Fish and Wildlife Service here at the National Conservation Training Center in West Virginia. We are excited to be co-hosting this series with the Monarch Joint Venture. Wendy Caldwell, coordinator for the Monarch Joint Venture, will be introducing today's speakers shortly. But first, if you have any questions during the presentation, please type it in the chat box. Most questions will be answered at the very end of all presentations completed. In about a week, you should receive an email with a survey link. It should only take about 5 minutes of your time, and your feedback is very important to us, and we would really appreciate it if you could take time to do that survey. I also want to mention, right after this broadcast at 3:30, NCTC historian Dr. Mark Madison will be interviewing Lincoln Brower, one of our speakers today. So please feel free to stick around. Okay, Wendy, are you ready to introduce the speakers?
Great. I will introducing our speakers, and thank you all for being here today. First, we're going to hear from Sonia Altizer, who is a professor in the Odom School of Ecology at the University of Georgia. There, she and her students study monarch behavior, ecology, and interactions with a protozoan parasite. In 2006, she launched a citizen science project called Monarch Health from her lab, and also maintains a webpage dedicated to monarch parasites. Lincoln Brower from Sweetbriar College has been involved in monarch conservation and research for over 60 years. He works in the fields of conservation, ecology, and ecological chemistry of the monarch butterfly. He's a distinguished service professor of Zoology Emeritus from the University of Florida. Last, we'll hear from Karen Oberhauser, who is the director of the University of Minnesota Monarch Lab. She's also a professor in the Department of Fisheries, Wildlife, and Conservation Biology at the University of Minnesota, where she and her students conduct research on several aspects of monarch butterfly ecology. In 1996, she and a graduate student started the Monarch Larva Monitoring Project, which engages hundreds of volunteers throughout North America. And now we're going to get into some really great content from these presenters, so I will turn the phone over to Dr. Sonia Altizer to introduce some really great topics.
Thanks, Wendy. So I want to start off by noting that the past 5 to 10 years have seen some really important advances in research on monarch genetics, evolution, and interactions with their parasites and pathogens. And today I want to briefly touch on some studies and advances that have come out in the most recent years. And a lot of this work is work that I've been involved with, but I also want to acknowledge several other people that have contributed a lot to what I'm going to tell you about today, including work by Jaap de Rode and his lab group at Emory University, work by Dara Satterfield, who's a PhD student in my lab, by Andy Davis, who's another monarch researcher, and multiple other people whose names you might see flash up in a few of the slides I'm about to show you. So I want to start out by thinking a little bit about recent studies on monarch evolution and genetics. And there's so much to talk about here, and there's some really good updates in the Monarch book that Karen is going to tell you about in just a little bit. So today I'll just mention 3 studies that I know a fair bit about and that have come out in the past several years. And all of this work collectively points to the fact that migratory monarchs in North America are unique and special from the perspective of genetics and the evolution of some traits. So the first study I want to mention was published 5 years ago, and this looked at the biogeography of monarch wings to ask whether migratory monarchs in North America have different wing sizes and shapes than monarchs in tropical and subtropical locations that don't migrate long distances each year. So we know that in migratory birds, long-distance migrants have longer, so they have bigger and more elongated wings, and the question is, do monarchs show the same trend? And so there was a study that Andy Davis and I did where we looked at hundreds of wild monarchs from North America and hundreds of wild monarchs from other parts of the world, including South Florida, Hawaii, Costa Rica, and Puerto Rico, where they don't migrate long distances. And we also raised some monarchs in what's called a common garden experiment in the lab to ask whether differences we see in the wild persist when you raise monarchs in the same conditions side by side. And the main result was that migratory monarchs in North America have larger and more elongated forewings. And this slide just shows an example comparison of monarchs on the left from eastern North America, migratory monarchs, and on the right from the Caribbean, just showing differences in the size of the wings and the shape of the wings. So these migratory monarchs had both larger wings and more elongated wings than monarchs that don't migrate. And so that was just one result. It didn't look at genes per se, but sort of at trait evolution. In terms of molecular genetic studies of monarchs, where researchers look at actual genotypes, a second study I want to mention was published last year by Amanda Pierce and her advisor Yap Dorota and colleagues, and they took several hundred monarchs, many hundreds of monarchs from locations all around the world indicated in this map in between 2007 and 2011. So these are all contemporary populations of monarchs globally. And they wanted to compare the genotypes of migratory monarchs in North America to non-migrant monarchs in the Pacific, in the Caribbean, and Atlantic islands, and in Central and South America. And they used 16 different what's called microsatellite markers. So these are sites along the genome that are not expressed into any traits. So these are silent markers that tend to accumulate a lot of genetic variation. And they can be used for genetic similarity tests. And one of the key results, and I'll show you this bit of an interesting looking plot at the bottom, shows how monarchs cluster genetically into 7 major groups based on these silent markers and the comparison of monarchs worldwide. And so the colors show combinations of genes that are characteristic of different regions. And at the far left are monarchs from the eastern and western U.S., and at the opposite end of the plot are monarchs, at the far right are monarchs from Hawaii, some Pacific islands, Australia, and New Zealand. And what this analysis showed is that monarchs cluster into 7 major groups genetically, with the groups that, as they cluster, indicated by the bar across the bottom of this slide. So North American monarchs do cluster, in this case, with monarchs to some degree from the Caribbean and from some Central American sites, and then there are several other major clusters of monarchs globally. So this analysis showed that there's a lot of genetic variation among monarchs worldwide. This same study also showed that North American monarchs are more genetically diverse than monarchs in other parts of the world. So these two graphs show one measure of genetic diversity for different monarch populations, with the left comparing monarchs in North America, which is the leftmost point that says USA, to monarchs from a whole string of Pacific sites. And as you go from left to right on this panel, you're moving away from North America and you're going further and further westward. So as monarchs move westward, genetic diversity goes down. And if we look at monarchs across the Atlantic, and again compare North American monarchs to those from non-migratory populations in Florida, the Caribbean, and across the Atlantic in Spain and Portugal, genetic diversity again decreases as you move away from North American monarchs. And then the third genetic study that I want to mention was published last year, which sequenced the genomes of about 100 monarchs worldwide from many of these same locations indicated in the map in the top of this slide. And at the bottom of this slide is an evolutionary tree based on all of this genome sequence data showing the relationships between all living monarchs worldwide. So the branches of the tree are color-coded to match the different regions of the map. And this analysis gave the surprising result that showed that North American migratory monarchs appear to be ancestral to all other monarchs worldwide. So the current population in North America gave rise to monarchs in all of these other places. And there were 3 separate radiations moving away from this central point, going to the south, which is the branch shown in red, through Central America and into South America, going across the Atlantic, or to the right, which is the purple branch, and then going to the west, which is the blue branch across the Pacific. And then the last, or one other really cool result from this last study that I want to mention actually compared the genomes of migratory North American monarchs to those of non-migratory monarchs elsewhere to see if there are any what we might call migration genes, and found that one gene in particular was different between migratory and non-migratory monarchs. So there's a rather nerdy diagram at the top of this slide showing the position of this gene. It was called the collagen type 4 gene. And North American monarchs actually lacked some information at this gene that resulted in their flight muscles having less collagen and therefore being more flexible than the flight muscles of non-migratory monarchs. And this same paper showed that migratory monarchs in North America have lower metabolic rates or lower metabolic demands during flight than non-migratory monarchs that were tested, and they were basically looking at how much oxygen the monarchs consumed while they were flying. And so what this means is that migratory monarchs in North America have a unique genetic mutation that allows them to fly more efficiently long distances than non-migratory monarchs. And I think another thing that this means is if we lose this migratory monarch population, we're going to lose this genetically distinct trait or set of traits that really allows them to fly these long distances. And we can't replace it by bringing in monarchs from other parts of the world. So now for the second part of my presentation, I want to shift gears and talk about some new insights from monarch pathogen interactions. And this is something that I know the best and I've been working on for the past 20 years, starting as a graduate student in Karen Oberhauser's lab and continuing on to the present day. And so what I'm going to tell you about are some monarch interactions with a protozoan pathogen called Ophryocystis electroscira, or OE for short. And this is an apicomplexan protozoan. It's a specialist pathogen on monarchs. This slide shows at the top how the protozoan spores appear at about 100x magnification, and the bottom panel has the arrows pointing to clusters of spores kind of clotted around the scales, the abdominal scales of a monarch butterfly. This pathogen is transmitted from adult monarchs to immature or larval stages. It's a very easy parasite to work with in the lab and in the field because it's very straightforward to tell whether monarchs are infected or not, and it's very tractable in terms of doing experiments. We know that this is a debilitating pathogen for monarchs. And monarchs that are infected tend to have difficulty eclosing from their pupal cases. They have shorter lifespans as adults. So this top left panel just shows that the probability that a monarch successfully ecloses from its pupal case decreases as the spore load of the monarch goes up. Infected monarchs have a lower probability of successfully mating. So mating probability goes down as spore load goes up. And as adults, they live shorter if they're infected with this protozoan than if they're uninfected. And they also, as a result, lay fewer total eggs if they're infected than if they're uninfected. So this has some pretty significant fitness consequences. And we also know that infected monarchs fly less well than healthy butterflies. And these are, there's a number of findings that have just come out in the past 5 to 10 years on this. There's a lot of genetic variation in monarch resistance to the pathogen and in pathogen virulence. So not all monarchs are equally susceptible to this pathogen, and not all pathogen strains are equally harmful to monarchs. And we've done a whole series of large cross-infection experiments where we raise monarchs in captivity and look at how they respond, how different monarch genotypes respond to different pathogen genotypes. And there's just a smattering of a few citations of work shown here in this slide where we've demonstrated this variation. And then some of this variation arises from different immune defenses that these butterflies harbor. So monarchs are actually pretty well defended, both immunologically against pathogens and chemically against pathogens and predators, as you might hear about in just a minute from Lincoln Brower. There's been a really nice set of work that's been done by Jaap de Rode and his lab that has looked at the role that the milkweed host plants have in the outcome of infection by this pathogen, and has shown, for example, that monarchs that feed on milkweed that's loaded with cardenolide toxins tend to emerge with lower spore loads, and they tend to live longer as adults than monarchs that feed on less toxic or lower cardenolide milkweed species which tend to emerge with higher spore loads, and they live shorter lives as adults. So these milkweed toxins don't cure the monarchs of infection, but they can lower the spore load somewhat and allow the monarchs to live longer. And then some follow-up work by Yap and colleagues showed that infected females preferentially lay their eggs on more toxic or more cardenolide-rich milkweeds as compared to healthy monarchs, perhaps to protect their offspring somewhat from this pathogen. So there's some nice work on the chemical defenses of these butterflies and how they affect pathogen infection. And then the last thing I want to note is just show you a few patterns of infection in the wild. So we've sampled many thousands, tens of thousands of monarchs over the past several decades for infection in the wild from different places all around the world. This is just a team of one group sampling monarchs at the overwintering sites in Mexico for infection patterns. And one thing that we found is there's tremendous global variation in the degree to which monarchs are infected with this protozoan. So this is a map from the forthcoming Monarch book. And this shows that monarchs in eastern North America, so each pie here shows, the black part of each pie shows the fraction of monarchs that are heavily infected by the protozoan. And eastern North American monarchs have the smallest slice of black in their pie, so they have the lowest infection rates. Also, we see low infection rates in monarchs in western North America. But some pies have a lot of black and very high infection rates, like monarchs in South Florida, in Bermuda, in Australia, New Zealand, and Hawaii. And these are all monarchs in parts of the world that don't migrate long distances. And so we followed this up with some work to ask, what is it about long-distance migration that might actually lower infection risk for these butterflies? And so there's a number of studies that have been published recently that show that long-distance migration actually allows monarchs to escape from habitats where protozoan spores build up over time, so it interrupts transmission each year. And it also weeds out infected butterflies because the infected butterflies migrate less well. And so we call these two processes migratory escape and migratory culling. And then the last quick result that I want to show you comes from results from citizen scientists in a citizen science project that we run called Project Monarch Health. And we engage a couple hundred volunteers from across North America in 25 different states and Canadian provinces, and this project has been going on since 2006. So these volunteers help us sample wild monarchs for infection, and then they mail the samples to the University of Georgia where we score them for infection status. And we've used this citizen science data to ask several important questions about the pathogen in recent years, and one is we've been really interested in how sort of the loss of migration or the formation of year-round or winter breeding monarchs in the southern U.S. might be affecting the spread of this pathogen and prevalence of this disease across North America. And here there's been some nice citations suggesting that there's a number of places along the southern Atlantic and Gulf coasts where monarchs now breed year-round and don't migrate. And that for the most part, these are happening in places where people are planting a milkweed species called tropical milkweeds. And so a graduate student in my lab a couple years ago recruited volunteers from the northern part of North America, indicated with the yellow dots, and from the southern part of North America, indicated by the blue dots, where monarchs were breeding during the winter months, to look at what's happening to infection in these winter breeding populations. And what she found was that the prevalence of this protozoan infection was 5 times higher in winter breeding monarchs in the two far right-hand bars than in migratory monarchs that were sampled in their summer breeding range at the far left and at their overwintering sites in Mexico in the middle. So this is suggesting that winter breeding really gives this pathogen a boost in terms of being able to be transmitted year-round. And the last picture that I want to show you is just what's happening over time to this infection across all migratory North American monarchs, or monarchs, east of the Rockies. And what we found is that the proportion of monarchs that are heavily infected has increased since 2002. So before 2002, prevalence was never above 5%, and average prevalence hovered around 2% of the butterflies being heavily infected. since 2002, the prevalence of infection has tripled in eastern migratory monarchs. And we're really interested in trying to figure out why that is, what's happening to drive infection higher. So I'll just end by thanking the many people that contributed to these studies. And this is just a small sampling of a much larger body of work to give you a flavor of some things that we've learned recently. Thank you.
Thanks, Sonia. That was fascinating. We have one quick question for you, which Karen addressed a little bit in the chat box. But before we transition to Lincoln, from Michael, from a population management perspective, would you want infected butterflies to live longer and therefore present a greater risk of infecting other butterflies? Could you expand on that? Karen provided a short answer, but I'm wondering if you could expand on that.
That's a great question. If infected butterflies live longer, that's a good thing for the individual butterfly from its perspective, but it's probably a bad thing for the population because those individual butterflies that live longer go on and transmit more disease than infected butterflies that have very short or truncated lifespans. And so what's good for the individual in this case is probably not good for the population. And so I would argue that that from a management perspective, that the answer is no, I don't think that would be a good thing.
Thanks. Now we'll transition to Dr. Lincoln Brower, who will speak on a number of other topics. So with that, I will turn it over to you, Lincoln.
Well, it's very nice to join everyone. That was a wonderful talk, Sonia. You've done so much in your lab with your colleagues. It's just fantastic. I wanted to talk about a few issues, mostly about the conservation issues that are surrounding the monarch. But before I get to that, I just wanted to give you a little bit of background. About 40 years ago, when I was at Amherst College, we did a study of asking the question, are monarch butterflies really unpalatable to their predators? And we trained blue jays in the lab to attack monarchs, and you can see that the bird is tearing off the wing, and then it tears off the legs and the antennae, and then it swallows the abdomen, and in about 5 minutes, the bird begins to look sick, and in about 12 minutes, the bird is vomiting off the other side of the perch. And what we found was that once a blue jay had had one of these emetic experiences, they would likely never touch another monarch as long as they lived. It's a very intense conditioning. And the question was, where are the poisons that the butterflies have in their bodies coming from? And it had long been known that they were probably coming from milkweeds. And so what we found was that we raised monarchs on 5 different species of milkweeds, and we then determined how emetic, or what vomiting strength would the monarchs have if reared on these different species of milkweeds. And the first one there is about 7.8 emetic units. In other words, a monarch reared on that plant, that species of milkweed, would have enough poison to make about almost 8 blue jays vomit. And then so on with lower and lower toxicity, and down to the plant on the right, which was a tropical milkweed, and we predicted it would be very poisonous, but it turned out to be not poisonous at all and contain none of the chemicals that cause the vomiting. And Sonia mentioned cardiac glycosides, and these are related to digitoxin, which is used in the treatment of human heart disease, and is a very, very interesting chemical. They're bitter tasting, they're emetic, and they will stop the heart dead completely. Well, with this chemical defense, when the monarchs were discovered overwintering in Mexico, one of the reasons we were desperate to find our way to the overwintering sites was because we wanted to see if the birds were eating the butterflies or whether they were chemically protected. And if you think about it, with these millions of butterflies concentrated on the Oyamel fir trees, if they didn't have some kind of a protection, they would probably be eaten by all kinds of bird and mouse predators. Well, when we went to Mexico the first time in 1977 in the Sierra Chinqua overwintering site, we found that 2 birds were eating monarchs apparently indiscriminately, the black-headed oriole and the black-backed oriole and the black-headed grosbeak. And the next image shows a monarch that had been discarded by the birds, and if you look at that monarch carefully, you'll see that it has no head on its body. So we asked the question, how many monarchs are being eaten by the birds? How effective is the chemical defense? So we made these nets that were 1 meter squared in diameter, and we set out 100 of them in the Sierra Chinqua colony. And then we collected the butterflies that were the birds that torn the wings off and dropped in the nets, and we were able to determine that the number of butterflies that were being eaten by the birds varied from a low of 3,500 to a number of 35,000 in a given day. Is the chemical defense effective? Well, yes and no. It depends partly on how much poison is in the butterflies, but the orioles and the grosbeaks, the oriole turned out to be able to reject the toxic butterflies, and the grosbeak was insensitive to cardinalides, so that they were affected. And if you look at that graph, you can see that there's a 6.8-day cycle in the graph. The birds come in, they eat butterflies, they begin getting sick, and then they stop eating them, and so on over a period of a cycle of about a week. Well, changing the subject now to the conservation issues, the monarch is an endangered biological phenomenon, and what that means is it's not an endangered species. What is endangered is this remarkable migration and the overwintering behavior, which is absolutely unique on our planet. And so the species is not endangered, but it's the behavior of the butterfly that is so remarkable is endangered. And the next slide shows passenger pigeons, which had a similar migratory phenomenon with millions and millions of birds migrating. And what is ominous about the passenger pigeon, which the last individual went extinct 100 years ago, is that their range of distribution is very similar to the monarch. And then if we go to the next slide, this shows the tabulation of the area occupied by monarchs in Mexico overwintering sites over the past 20 years. And you can see that it's going down, 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 down. And the last 3 years are all-time low for the 21-year period. And there were even more monarchs there before these records were taken. So one of the very likely reasons why monarchs are declining, especially in the last decade or so, is the development of genetically engineered plants which are resistant to herbicides. The genetically engineered corn, genetically engineered soybeans are the chief culprits, so that these plants are resistant to herbicides, and they're planted in the corn, corn and the soybeans are planted, and then they're sprayed. And the next slide, you can see a picture I took a year ago in Kansas. You can see a monoculture cornfield on the left, monocultured grass in the middle, and monoculture soybeans on the right. And the next slide shows corn looking at the base. And if you look at the next slide, you can see just where there's absolutely nothing growing. So what happens is that the soybeans and the corn are resistant to the glyphosate, this Roundup herbicide, and it kills everything except the corn or the soybeans. And the problem is that, as Karen has shown in her lab work over the years, and along with others, is that monarchs are feeding on milkweeds that are abundant in the cornfields, in the agricultural habitats. Well, now that whole habitat has been eliminated. Milkweeds have been killed off. And if you look at the next slide, you can see a monoculture soybean rows with no weeds growing. And then we did an experiment in the next slide at Sweetbriar College this past year. And what you see here is a field which was sprayed with herbicide about 2 or 3 weeks before this picture was taken. And it's sterile, it's a desert. So these herbicides kill everything. They kill the milkweeds, they kill the nectar sources. And the next slide shows a milkweed that is serving as a nectar source for the monarch. And the next slide shows goldenrod. And if we go to the next slide, we've looked at the relationship of nectaring, the way in which monarchs derive enough energy in order to migrate and get through their complicated life cycle is they convert the sugars that are present in the nectar to lipids that are stored in their body. And what you can see here is that the lipids go up. They're very low during the summer, but as the butterflies begin migrating to Mexico, they build them up, and as they head through Texas, they increase their lipid contents by 500%. They're literally butterballs. So one of the things that we did 2 years ago with Kip Kiphart and colleagues was we went out to Texas at the height of the drought, and we asked the question, the drought is affecting the wildflowers, and how is that affecting the lipid contents of the monarchs? And there you see a springtime in a normal year with the wildflowers growing, and then at the height of the drought, the year before that, was just unbelievably dry. And the Medina River in the next slide was flowing during the normal season, but during the height of the drought, there was no water in the Medina River at all. So we collected monarchs in the next slide, and we looked at their lipid contents, and this is during a normal series of years, no drought, and the average is pretty high, about 80 milligrams of fat. And then this in the middle was the lipid content of the butterflies during the drought, and about half as much as during the normal period. And we had a control series which was very lucky. There was a big liatris garden that was in bloom and had been watered and had normal lipid nectar contents, and the butterflies that were feeding on those flowers were normal. Well, changing the subject again, one of the problems that I noticed on my first trip to Mexico was logging that was damaging the overwintering site. And I got together with Mexican colleagues and American colleagues, and we found that there was a 44% decrease in the quality forest over a 25-year or so period. And that is due to logging, which is still going on, and here you see it. It's going up, and around 2007, there was massive illegal logging in the overwintering area. It's dropped off now. The Mexican government has made a major effort to stop the illegal logging. But just to show you examples of it, here is the Sierra Chinqua, and you can see that the area where the butterflies would normally be sitting on the trees has been stripped of its trees. Another area which is stripped, and another area taken from aerial photography that we were able to do. Another major area where the butterflies land and overwinter, stripped. And this was very, very serious impact on the whole overwintering region. And the next slide shows Cerro Pelon on the south face, which has not been logged, to give you a contrast of how beautiful the area was. Now, if we just go over the top of that mountain down to the other side, we'll see horse logging going on. And the next slide shows the horse loggers relentlessly working, and the next slide shows an aerial photo of the west face of Cerro Pelon, which has been stripped of its trees over the past 20, 30 years, and that horse logging is still going on in spite of the statements that are coming out of Mexico to say that it's not. We must stop this low-scale logging. The next slide shows the same area, and it shows that where the colonies have been noticed in the past, and in fact, it was on the west face of that slope that Kathy Brugger and Ken Brugger discovered the first overwintering site on January 2nd, 1975. So the next slide, just wrapping up, the monarch butterfly situation is an endangered biological phenomenon because of habitat deterioration in Mexico, which is coming under control. That's the good news. But the bad news is chemical agriculture is eliminating the food plant over such huge areas so that the monarch has truly become an endangered biological phenomenon. And then finally, this is something that is so beautiful we must save it. And that's what much of the research that we're all trying to do is keyed to build a case why. And in fact, there are huge efforts now made to save the monarch butterfly, and we need to get everybody involved in this effort of planting milkweeds and protecting the forests in Mexico. Thank you all very much.
Thanks so much, Lincoln. That was great. I've got one question from Steve coming in here. Could you expand on your comments about populations being higher prior to the 1990s monitoring data?
Yes. Well, the problem is we developed a method of monitoring the area about 15 years after we really got started. And in the early days from 1975, 76, 77, on through the 80s, the colonies were bigger and there were more of them. Several colonies have actually been eliminated, and one of the really frightening aspects is the Sierra Chinqua, which in the early days had 2 to 3 hectare colony sizes. The past 3 years have been tiny, like a tenth or 20th of what they originally were. So I'm concerned not only about the general decline, but the loss of some of the overwintering sites altogether.
Thanks, Lincoln. With that, I'm going to transition to our final speaker, Karen Oberhauser.
Yeah, so thanks for that introduction and for all the questions that are coming in here. I was thinking as I was listening to Sonia and Lincoln that the three of us really represent 3 generations of researchers, and it's so exciting to be part of this group. I mean, Lincoln has been doing this for a really long time, and Sonia and I are kind of latecomers, but we really have learned a lot from each other over the years, so it's a lot of fun to be here. And we're all working hard on training the next generation of monarch researchers as well. So I'm going to go kind of quickly here so that we have plenty of time for questions. So Wendy will be collating the questions that come in, so keep asking questions in the chat box. And I'm going to bring us kind of back down to the breeding season and think a little bit about 3 topics that I've been working on a lot recently, and that's natural enemies, not focusing on OE because Sonia's the expert on that, but also thinking about climate change and population dynamics. So we'll start with natural enemies, and I've been really interested lately in trying to figure out how many monarchs survive. Say a female lays 500 eggs in her lifetime, how many of those actually turn into adult monarchs? So this is kind of a review of past literature, so I'm going to kind of combine new stuff and old stuff here. And what you can see on this slide is that there are very low rates of survival. And the first person to study this was Sue Borkin, who went out and surveyed 1 site. She published this in 1982, and she found there was about 12% survival from egg to 5th instar. Then there was a study that was published in 2001 that involved people all over the United States looking at different kinds of habitats. And we found, depending on the habitat and the location, survival of about 5 to 15% of egg to 5th instar. And then a couple more studies that were published in the 2004 book, one by Michelle Prysby, found 12 to 19%. Bill Calvert had one down in Texas with very low survival. He was looking at places where there were fire ants, and he found that the fire ants basically ate all of the caterpillars. None of them survived in some sites. So he actually found 0% survival. So there's been a lot of work done over the years with people trying to understand this. A lot of people have also looked at different causes of mortality. So we have reviews of all of the different things that people have recorded eating monarchs and also the different parasitoids, and those are in a review paper that's just out in the new book that's coming up, that just came out. So here you have some pictures of a lot of the different things that eat monarchs, from the one with the microscope in it,
or the little hand lens looking at a little tiny spider mite eating an egg. You have wheel bug eating a second instar larva, and all sorts of mortality. And on the left is the tachinid fly, the maggots from the tachinid flies. So lots and lots of things eat monarchs and cause all of this mortality. So I wanted to just briefly describe a study that one of my students, Alma Deonda, did, and this is published in the upcoming book. And we actually tracked where all the other studies looked at, kind of counted the number of eggs they saw, the number of first instars to get mortality. Alma actually tracked individual eggs. So she was at one site, she did this over 2 years. So this graph shows compiled data. And it starts with, here, I think I can get a little arrow. No, maybe I can't do that. Anyway, it started if you have 100 monarchs laid on one day, a little over 60 of them are alive on the next day, and then we go down to about 40 of them. And she found about 60% survival, so that means 40% of them die every day, and this happens over the egg stage, which are the first 4 days on this graph, and the first instar and the second instar stage. So in this study, to the end of the second instar, we only had about 2.5% survival. And again, this is just one location, but it's actually tracking individual monarch eggs through their development. We had to stop tracking them because when they molt into third instars, they often move off of their natal plant. And then this is some new information that's just in press right now. It's not in the book, but it's in a journal article that's coming out in the Journal of Animal Ecology. I'll put in a plug. There's going to be a great journal edited by Andy Davis, which has a lot of articles taken from Monarch citizen science projects. So this graph is showing findings from the Monarch Larva Monitoring Project. And it's showing you over all of the years of this study. So the Monarch Larva Monitoring Project started in 1997. And we have volunteers monitoring in a lot of different regions. On this graph, I'm just showing you the data from 3 of the regions, or actually 3 time period region combinations. So the south in the spring, so I see Kippard is here. A lot of the data represented by this dark black line come from Kip and other people that he's trained down in Texas. And this is survival. This is the number of fifth instars people saw divided by the number of eggs that they saw. So we're getting survival from egg to adult. So you can see it's really variable from year to year. So this demonstrates the importance of long-term studies and really understanding the dynamics of monarchs. You can see the north in the summer. So the gray line on this graph is showing you what's happening. These are actually the monarchs that will be the ones that migrate to Mexico. Lots of variation from year to year in survival. And there actually is a significant downward trend with the survival information that we're trying to understand. We don't know the reason for that at this point. It popped up again in 2014, but over time there is a significant downward trend in these data. And this is just a paper. Obviously, you're not going to read all of the, or this table, you're not going to read everything on here, but this is a review of the literature that we did for this paper. So we've pulled together all of the different studies that documented predators and parasites of monarchs. So this is kind of a fun compilation of the data, and you can see a lot of people have studied egg mortality, larval mortality, fewer studies on pupal mortality, but a few of them, and then quite a few studies of our adult mortality. So this is kind of fun to see all of this. Okay, now switching gears here, another thing that I've been really interested in is how climate change might affect monarchs, and I used one of Lincoln's pictures here that you saw in his presentation showing really what can happen during a drought. This is monarch habitat during some years, but obviously not the year that this was taken. So we've done a lot of thinking about how a changing climate might affect monarchs. And I've listed some of these things here. Certainly extreme climate events like the drought picture you're seeing in this slide, but also winter storms. We've heard a lot about winter storms killing monarchs. Another thing that might happen with temperature shifting is that the location of optimal habitat for monarchs might change. So for example, if we have increased temperatures in the breeding range, will monarchs move further north than they currently do? Or if we have increased precipitation in the wintering grounds, what will that do to where monarchs should be during different seasons? Another thing that we're doing a lot of work on right now is looking at the interactions that monarchs have between plants that they depend on and natural enemies. So thinking of the plants, will climate change affect the range of milkweed? Or will it affect the range of the oyamel fir trees that monarchs use during the winter? Or will it change the timing of all of these predators or these parasites that I've talked about? So how are these interactions that monarchs are going to have with everything else in their environment going to be affected by climate change? And finally, we're thinking a lot about what might happen with migratory patterns. Depending on the importance of different stages of this migration being driven by temperature, it's possible that climate change could have an impact. So, so these are kind of the things that I just mentioned were indirect effects that could be changed, but also we know that low temperatures and high temperatures affect monarchs directly. So Lincoln and his students did really great work showing that monarchs are very susceptible to low temperatures during the overwintering season, that they'll freeze to death, especially if they're wet. Low temperatures can make them more vulnerable to predation. High temperatures can make them use up all that fat. Lincoln mentioned that they're little butterballs when they get to the overwintering sites, but if it's too warm, that lipid can be burned off. It also might make their diapause, the arrested reproductive state that they're in when they get to these overwintering sites, end prematurely. So there are a lot of potential direct effects of climate change on monarchs during the winter. During the breeding season, there are also lots of possibilities. So this March to October period when monarchs are up here in the United States, we know that low temperatures and rain both lead to slower development. That makes monarchs more vulnerable to predation, which we've already learned is really important. So here you have this little field bug nymph eating a second instar. High temperatures we know lead to shorter lifespans, higher mortality during development, and also smaller monarchs. So we've done some studies here looking at the effect of size on monarch reproductive ability. So there are a lot of potential implications of temperature for monarchs. Dry conditions will affect the milkweed. So lots of things going on. And some kind of interesting recent research that's been done potentially linking changes in the monarch population to climate. As Sonia mentioned, we have evidence that monarchs are staying in the southern United States for the winter. Several different studies have shown that. And Sonia's student, Dara, showed that more monarchs staying in Texas during the winter time might lead to increased OE infection rates. Or it could expose monarchs to freezing temperatures if they lay a lot of eggs in the south and then an unusual cold spell comes. So we've been really focused on thinking about how precisely does temperature affect monarchs. And there are a couple of studies that are coming out in our new book. We found that monarch eggs and caterpillars can also actually survive very cold temperatures. These are temperatures in Celsius. Eggs can get down to minus 25 degrees C before they die. Larvae down to minus 10 or 13, depending on what stage they're in. Minus 18 for the pupae. So they can survive very cold temperatures. So we don't think that cold, breeding in the south, that cold temperatures will really hurt them because it rarely gets this cold in the south. And larvae can also withstand bursts of high temperatures up to 40 degrees C, which is about 104 degrees Fahrenheit. But if it does get this hot for them, their development slows way down. So here are some direct effects on monarchs during their breeding season of extreme temperatures. And finally, I just have a couple slides to kind of tease you a little bit about some current work that's going on about population dynamics. As you all know, there's a lot of attention from a lot of people on monarch conservation right now, so we're very interested in where we should concentrate our conservation efforts so that where they'll make the most difference. So there are a couple different kinds of research projects that are going on to address this question of targeting our conservation efforts. And one is looking at how monarch numbers at one stage correlate with those at subsequent stages. So the thinking here is that if we have monarchs at one stage, say breeding in the south, and there's not a strong correlation between the numbers we see breeding in the south and the numbers we see breeding in the north, that means that something that happens in between, like during their migration, if we use this from the breeding in the south to breeding in the north, the migration from the south might be driving if we don't see a correlation. So that's one kind of study, and I'll show you a slide about that. But first I just want to introduce another way that we're looking at that. And this is if we use actual data on vital rates in monarchs, like how many monarchs survive, the kind of data that I was just showing you, or how many eggs they lay. And what happens if we change those vital rates in population models to see how different stages during this annual migratory cycle affect population growth? So both of these methods are modeling. This isn't actually going out and collecting data. There are two different ways of modeling the importance of different regions to monarchs. So this figure is from a paper that's in our new book, and it's a really complicated map, but it's showing all of these different stages of monarch migration. So for example, the one on here represents monarchs coming up from Mexico, coming into the southern United States. The two represents them breeding in the south, and then the threes, because we've got a 3A and a 3B, represents them migrating up, and then they breed. So we looked at correlations along all of these different steps, and what we found is that this spring recruitment stage, or this breeding stage that's represented by the two on this map, really has a critical influence on the size of the breeding population. So what goes on in the southern United States is really, really important. We've also found that there's not a very strong correlation between summer population size and the winter population. So we think that this fall migration, which is represented by the dashed lines and the circles with the 5A and the 5B in them, might be really key because we're not getting this correlation between breeding in the north and the winter population size. So this is one way to look at how do we target population, or how do we target conservation, as we look at what might be driving the population. And another way, there's a study published on this in 2014 by Tyler Flockhart in Canada, and another one that I'm working on with several other people, some of whom are on this call, I noticed, using slightly different methods, but basically we're using our best guess for vital rates during all different stages of the monarch and seeing how tweaking those vital rates at different stages of the population drives population numbers. And both Tyler Flockhart and the preliminary data that we're finding in our work suggests that increasing the success of the population during the breeding stage of the life cycle will have a large effect on population size. So lots and lots of work being done with really specific conservation targets, trying to figure out how we should best preserve monarchs, which Lincoln argued very succinctly is really important to all of us. And I just want to end so that we have plenty of time for your questions. I just want to put a plug in here that Sonia and I and Kelly Nail, who's a graduate student here at the University of Minnesota, just edited a book, and I've listed all of the names of people. I'm sure a lot of you are on this call. All of the people listed here contributed to this book. So this is available now from Cornell University Press. It summarizes a lot more research than we've been able to talk about today. So I really look forward to talking with people about this research and hearing your questions about what's going on. So thanks a lot for your attention, and hopefully we'll have a lot of questions that Wendy can ask us.
Thanks, Karen, and thanks, Sonia and Lincoln. I learn something new every time I hear you all speak. So thank you so much for sharing your expertise with the group today. For everyone still on the line, looks like we've got quite a few of you. We're going to transition to a question and answer period. I'm going to start with a question that came in from Les, and I'd like to get our presenters' take on, maybe Karen, you could start to address this topic, but your take on the scale of the restoration efforts that need to occur for monarchs. So you talk about targeted restoration, but could you talk more about really what it is that we're looking at? And the original question was about milkweed plants in someone's backyard, and can growing milkweeds in your backyard really make a difference for monarchs, or what is the scale that we're looking at for restoration?
So that's a really great question. The target, the Fish and Wildlife stated target for monarch conservation is 6 hectares. So Lincoln showed you a graph of the size of the overwintering population. It's down around 1 right now. So we want to have 6 hectares of land, and that's approximately 225 million monarchs. There are a lot of different estimates of how many monarchs are in a hectare, but we figure, around 30 to 35 million monarchs per hectare. So there are a lot of ways to think about, what our targets are. And one thing we can do is say if we want to add, say, 200 million more monarchs, one way to think of it, we've done some estimates using MLMP data, and I didn't talk about this today, but on average, given all of that mortality, you only get about 1 monarch off of 30 milkweed plants because of mortality. So if we multiply, we can kind of multiply, we want 200 million more monarchs, and we're only getting, 30, you need 30 plants to make a monarch. We're talking in the billions. We're talking billions of milkweed need to be planted. Obviously, that's kind of a scary big number, but I think right now what we need to think about is we basically need to plant milkweed wherever we can. And the nice thing about monarchs is that milkweed, that they can find fairly small habitats. So, planting a couple milkweed plants on average, it's always better to plant more. But if all someone can do, if they only have space for a few, that's better than nothing.
Great. Sonia or Lincoln, anything to add to that?
Well, one mitigation factor that we've talked about is getting the highway departments, the edges of roads, to stop spraying them and to manage them for wildlife to a greater extent than they have, and also power line right-of-ways and electrical gas line power. So there's a lot of area. There's something like 4 million miles of roads in the eastern United States. And if we could protect a substantial percentage of those roadsides, it would probably be a major contribution to monarch milkweed biology.
Great. Karen, could you expand more on, from a conservation management perspective, on the lack of correlation between the size of the summer and overwintering population sizes?
Yeah, and that's kind of a complicated thing to think about. So this correlation is looking at across all of the years for which we have data. We don't see a strong correlation. So that's not saying that habitat during the breeding season isn't important. It's just saying that some of the factors that drive population numbers on a year-to-year basis, so that would be things like all of that variation that we saw in predation or parasitism, that survival during the migration is key. So I didn't mean to imply that habitat wasn't important, but just that the kind of the little tiny things that affect monarch numbers coming out of the northern breeding range might not be as important as the things that happen during the fall. Another way to think of that is it might be that we have more consistent numbers, so that from year to year we get consistency in the northern breeding range, and that there's a lot of variation in in migratory survival, and that's something that's very difficult for us to measure. So it's just saying that the annual variation is not driving things. It's not saying that the actual amount of breeding habitat is not driving things. I hope that was clear. I felt like I was kind of convoluted there.
Well, if anyone has a follow-up question to that, please put it in the chat box. If not, we will move on. Lincoln, I'm gonna start with you on this one, and others feel free to jump in. Could you address how migratory and non-migratory populations interact in regions, such as the Southwest, where both might be present?
Well, the one population that I've studied, and Karen and Sonia have also visited this population, was near the Miami airport. And the butterflies, it was a wonderful, wonderful sight there. There was Asclepias incarnata and Asclepias curassavica growing, and another milkweed vine. And so we were able to compare monarchs and queen butterflies feeding together. But what is I think the most important thing is what Sonia's already talked about, and that is that these residential populations that do not migrate have high levels of infection of OE. And one of my concerns is the planting of milkweeds, indiscriminately taking the tropical American milkweed and planting it in further north, because it's so very attractive to monarchs that they lay their eggs on it and go through more than one generation. And if they're breeding on the same plants for more than one generation, the probability of infection rate is higher. Sonia, what's your take on that?
I think the potential for interaction is really high, but I think it's something that we need more information on, and so hopefully in the next year or two there will be more information from our lab and from other research groups on to answer several questions. And so one question is, how common is it for fall migrant monarchs, when they pass through sites with non-migratory monarchs, to abandon migration and join the resident breeding populations? And it seems like there's some evidence out there that suggests that the potential is there, but to really try to get a better handle on the probability with which that happens seems important. And then to what extent do monarchs that are migrating north in the spring out of Mexico interact with winter monarchs at these winter breeding sites in a way that might lead to spillover of disease from high prevalence areas where monarchs have been breeding all winter to native milkweeds in places like Texas, and we just don't know the answer to that right now. But again, I think the potential is there, and so trying to get I think, I'm working on this with Dara Satterfield, and I think some other people will be working on this question too. So we'd really love to know the answer.
Thanks for that. Sonia, I'm gonna start with you on this next question, and maybe Karen, you can weigh in, and Lincoln as well. But could you talk a little bit about your perspective on the value of rearing and releasing monarchs?
Wow, that's a really interesting question. I think there's many different ways to answer that. I think that, in terms of what I personally think, I think there's a lot of value in terms of education and outreach, and really getting people, there's nothing like seeing a monarch at close from its chrysalis up close to get people to care about these butterflies. And so I think just being able to watch them really closely, watch their development and marvel at that is so important. And rearing monarchs, for some people, seems to really help serve that role and just capture the magic of the butterflies. I don't really know if I would feel very positively about the conservation value of captive rearing as a way to supplement or support the population, partly because when we take monarchs out of the wild and we bring them indoors to rear them, we're bypassing a lot of natural selection that would be happening in the wild. And that bypassing process might not be a good thing. It also seems that there's the potential to support disease transmission if people are rearing monarchs in a way that fosters when you keep these animals in close quarters at high densities, it really can foster the transmission of infections. And so if we don't know what we're doing and we're not using best sterile practices, it really can lead to unwanted disease transmission and the unintentional release of infected monarchs into the wild and augmentation of levels of disease. So that's not a good thing either. And then I think it might, what we really want is sustainable population of monarchs in the future, not a population that depends on people to coddle them and rear them and release hundreds and hundreds and thousands of them every year just to keep the population going. And so it seems like focusing on habitats and what's happening to monarchs in the wild, I think, is a more valuable place to focus our efforts and energies than on rearing. But that's my opinion.
I'm going to jump in just quick. I really loved what Sonia said there, and I'm going to add one other thing to that, is that when people rear monarchs, when they collect them from the wild and rear them, I think that it has a lot of educational and connection value, but it can have some scientific value, and that is that the Monarch Larva Monitoring Project and Monarch Health both have ways that people can report valuable data on monarchs that they collect and rear from the wild. So a lot of our data on understanding monarch populations comes from data that people have reported. If they rear an egg, does it survive? Does it turn into a monarch? Is it parasitized? Does it have OE? So we can really learn a lot from every single monarch that people rear. So I would encourage anyone who's rearing them to find out about how they can contribute data from that work that they do to these two citizen science projects, and their data will be used to help us address monarch conservation.
Thanks, Karen and Sonia. That was a great response to that question. There were a few questions on roadside habitat, and I'm not sure which of you is the best person to address this, so whoever feels equipped, please jump in. Do you think that drawing monarchs to roadways and roadside habitat, will that result in a lot of deaths of those butterflies being hit by cars?
So I can jump in here, and then Lincoln, you could jump in too. Yeah, probably some of the monarchs that grow up along roadsides, if they've laid on a milkweed plant on a roadside, they might get hit by cars, but probably the net benefit of those roadsides would still be positive. If that's the only habitat available in a big area, and the monarchs would otherwise have to fly long, long distances before they can lay their eggs, and we know that can decrease fecundity, or the number of eggs that females can lay, that the roadsides are better than nothing, in my opinion. So
well, the other thing is that if you go out into the Midwest, in the farming community country, that there's hardly any traffic on those roads at all most of the time, and so that I think that that protecting those roadsides, and botanists looking for wild plants drive along, looking out the window, and find all kinds of wildflowers. It's so that it is a very important native habitat that's remnant. And if we made efforts to protect it by not mowing them all the time and by not spraying them, I think it would be a major contribution to monarch fecundity.
For a slightly different perspective, I'm sure some people are aware of the study by Steve Malcolm and May Berenbaum and colleagues, published in 2001 on mortality of Lepidoptera along roadways in central Illinois. And in that study, they went along roadways in the vicinity of Champaign-Urbana, Illinois, and they estimated that the number of monarch butterflies killed during the course of their study could have been 500,000 individuals during 1 week in the state of Illinois. And so that seems like a big number, and it would be really interesting to take a look at those data and to try to get more data on road kills, because it seems like the answer to the question really is probably something like it depends. It depends on the speed of the road. It depends on the other roadside management practices, exposure to chemicals, and there are probably a lot of roads where the monarch mortality is really high, and then some roads where it could be a source for the population.
Well stated.
Yeah, thank you for all of your very intuitive answers for that. We have to wrap up, so I think I'm going to leave our question and answer period at that.